All right, grade 12. Our thomeng ka this topic ya DNA. DNA as code of life. Now, what is the most important thing is something re hopole iso. Ntho tshwantseng we hopole is that we need to start way at the back and give ourselves a revision ya a cell before we get into uh, DNA. Remember, cell is this one. Let's use this one. Remember, normal animal cell is this. Aha, got it. Remember, an animal cell consists of, for example, your cell membrane. Cell membrane. Your cell membrane it controls the activity. It controls traffic. So what in cell e control again cell membrane it's only border control well not come South Africa but control is sip but what like so it control what enters and leaves the cell, then that's the cell membrane. And then there's a lot of other organelles like your endoplasmic reticulum or your Golgi body, mitochondrion or mitochondrion something by it mitochondrion ke la fang energy ke escom ya ka mo seleng, but this one is the private school escom wa bona e phela ile constant ka go fa energy so long as when we fa the sugars and oxygen needed to perform that function. And then we'll have, if this was a plant cell, we'll have something called the chloroplast. Chloroplast is di solar panel tsa cell. The light converted into energy for plant cell. What energy is that? Glucose, chemical energy. And then innerly, what we call the nucleus. A cell has what we call the nucleus. Nucleus is the nuclear membrane. Nuclear membrane remember nuclear membrane in a pores ke masoba ke ka mo di RNA di tsenang di tswa and anything else ne. So you got the nuclear pores, and then it's a double membrane. Remember, it's a double membrane. And then ka re tsena ka mare ho na le nuclear plasm. Nuclear plasm ke that jelly like substance ka mare nucleus where you find your DNA ne. And then tse nka ditlhalenyana tse ke yona my lines tse ka ditlhalenyana tse ke yona DNA ya hao in the form of what we call chromatin network. Ke DNA ya hao, it's an organized DNA e thoma sele area divider, it's going to wind up yo tatagana ira what you call a chromosome or it's chromosome ke tsela tse bjana tse kwa ditsebenyana. So these are chromosomes. We usually see them like this. Hey, my art skills are horrible. Yeah. But in this time it's an organized. It's a chromatin network. Remember this from grade 10. And then we have this other part where it's called the nucleolus. Nucleolus that is inside the nucleus. So in a nutshell, this is a revision of the cell. I just wanted you to know read a revision quickly. Yeah, cell before it's entered into DNA. So lesson ya DNA is divided into what? We divide as so. DNA. DNA re tlo lebella re ke what is DNA ne re fetsa ra go history ya yona go tlile bjang gore re itse ka di DNA on or no Thomas story se sena history or no le history re tlhaka and it's interesting yana tse tsa teng. And then ra go ntwenyo ra ka mo structures sa teng gore ke mang pila from based on this history beno re tlo ya re ke mang pila tlile ka structures teng ba re ke double helix. And then re tlo hapo, now that we know what is the structure sa DNA, re tlo ya ko location, GPS, location ne re tlo itse re DNA re kry-a kae ha e kry-we ka mara nucleus fela le ka mo mitochondrion teng wa bona. and then the role looking e playa role e mo more. So re tlo lebelela role e. So starting then, we have a lot to go through. What is DNA? DNA basically is a nucleic acid. DNA is nucleic acid, and we have two types of nucleic acid. Remember in return, already it's what we call organic compound. So DNA it's an organic compound. It's an organic compound, and it gets a two these types of nucleic acid. Remember, really like DNA, RNA. So now that you more great to are building in food, repeat a deoxyribonucleic acid, and then a very ribonucleic acid. Di differ ka structure, right? So RNA is single strand is so it's a single strand, and A is double helix, ne? Double helix is so double helix is the structure is so. Here A is DNA is double helix. It's when there are two strands it's radibia together, and then radi twister so. So look at step later. So I'm not looking so was twisted one. It's only DNA, and then RNA is one half of step later saha. So that's basically what it is. So since we know what are these nucleic acid, everything. Remember go return really the building block. Don't something again. So nucleic acids di the monomers. The monomers. The monomer can get the building. It's only the DNA again. I don't know it's a DNA building blocks. So the monomers that the nucleic acids. The monomers for nucleic acids are divided the nucleotides. The monomers of nucleic acid are the nucleotides, and the nucleotides again we'll get into that just now. So nucleotides they are different. It's three. 1, 2, and 3. Ya ntlha nucleotide e forma backbone ya DNA ya gago ke sugar molecule. E rile ka pentose sugar molecule. Usually re draw into so. Pentose meaning it has 5 sides. Then the second one is phosphate group. Phosphate group meaning it rile ka di phosphate. One phosphate is somewhere there. And then there is 3, which is the most important. Ki nitrogenous base. So ki nitrogenous base. So re tla tsena ko structure sa yona just now. Now that ra itse gore building blocks tsa nucleotides meaning re ra DNA le RNA ke di building blocks tsa nucleic acid, sorry, ke di nucleotides, and ke sugar, sukiri e, phosphate le nitrogen base or nitrogenous base. History yeah, all of this. Who discovered DNA? Ka 1952 ne no na le ana mamazala bare Rosalind Franklin. Rosalind Franklin daya mamazala ke yena a discoverileng this DNA structure, ne? The double helix, but a berekisa X-ray. She bombarded a whole lot of energy more X-ray structures are high, and then I create what we call the image, the first image ya DNA. Then NASA berege Ali 1. I need my slides quickly for this history. NASA berege Ali 1 during this time. During this time, America let me see if I get it quickly. during this time a bereka le Maurice Wilkins ka 1952. Now Maurice Wilkins a ka wa itse nako ela basadi ne ba sa tseiwe serious dimanes ne Rosalina a tla ka image ya ntlha ya DNA kandi ko saiteng Waston le Crick le bona ba nyaka structure sa DNA o ka re ke mpša ya nna Judas a no tseya image ela e bontsha diamonds evan khane structure sa se bjana ba berekisa image ya Rosalind Franklin and then ba formulator what we call the 3D double helix model for DNA ebe ba tswe so after that mosadi o aswa Rosalind Franklin aswa after that bona ba tswella pele in 62 ba winner the Nobel Prize for discovering the structure of DNA. Maurice Lena revised an award for his X-ray photography. Well, near Rosalind Franklin, but we use it. So, we double say, I ko Waston and Creek, by Spanisa, if it's a winner, Nobel Peace Prize, 62. So, that is the history of DNA. Great. Now, Arwele, really level history and then level the structure of DNA. Are concentrating more DNA. DNA re ke double helix. What is a double helix? Double helix basically it's one of the two step ladders. One step ladder if it's a way we twist. It looks like this. This is the double helix of the DNA structure. Double helix. Yeah. Now, remember I told you DNA is made up of nucleotides. So what are these nucleotides? Now are the drawn. Nucleotide first and foremost, we have a pentose sugar making up this DNA. A pentose sugar connected to a phosphate group. And then a nitrogenous base. So this is the phosphate group connected to phosphate group, and then this is the sugar, and these two phosphate le the sugar ke backbone ke mokokotlo wa DNA ko morago. So basically o bona line e so is basically phosphate le sugar, and then e botla nitrogenous bases. The nitrogenous bases ke tsone tse di bona mo hare ha di backbones ke tsone tse di mo hare ne nitrogenous base di kopana. Now di kopana bjang? Remember nitrogenous base now let's go in detail. Nitrogenous base now let's say four types tsa nitrogenous base. Your number one king, number one we rebitsa guanine. Number two ki cytosine. Ne? Then number three, there's a reason why ki kwalaka different colors. King ki thymine. Number four ki adenine. Now, why ki kwalaka ka colors outside? These two re di bitsa di base pairs. Again, le guanine and cytosine le tsona ke di base pairs. Ke ra reng he ke re ke di base pairs, meaning guanine e tlo phela e bonda le cytosine ka di weak hydrogen bonds. Thymine e tlo phela e bonda le adenine ka di weak hydrogen bonds. Ne? So for your information, we represent ka di capital letters. E over G, capital letter G, e over capital letter C, e over capital letter A, and e over capital letter T. So whenever you see T,
with your bondaling li A. That is in DNA only. Ne? In RNA, thymine and adenine, they are completely different, but that's most stories. Ne? Ho bona A, or your bondaling li T. Fela. Alternatively, ho bona G, guanine, your bondale C, got the weak hydrogen bonds. Ho bona cytosine, or your bondale guanine. Ne? And then repeat the base pairs. Ne? So this is the structure of DNA. Ne? Good. So that's basically what we need to know as a form of structure sa DNA. So are we to the slides? Are we to the slides? Let me exit here. Good. So this is basically your double helix structure. So I wanna, if for example this is thymine, it will bond with adenine. If this is cytosine, it will bond with guanine. Vice versa, guanine, cytosine, so forth and so forth. And then just like I said, the phosphate group le di-sugar molecules tsela di form a backbone. So basically at the back here, the backbone is every DNA structure ke the phosphate group and nitrogen and sugar molecules. Ne? And then obviously we call it the deoxyribose sugar. Ne? Good. As already explained, Ilori, structures are nucleotide severe. Now, are yung location GPS re kira ka DNA. Remember ka mo seleng ya hao re boletse re ka mara nucleus ya hao ho na le DNA. That DNA re fa sefane bjanong e kreiwang ka mo nucleus ke nuclear DNA di tsamaya ka sefane, ne? And then e kreiwang ko ntle as the main DNA ka mara nucleus ya hao e controller all the activities of a cell. Then e kreiwang ko ntle ha nucleus gore e extra ke extra re kira extra nuclear outside the nucleus. So that means re kira outside the nucleus. So we call it extra nuclear DNA. And where particularly re kira go chloroplastin, ne? Chloroplastic DNA and then mitochondrial DNA. Mitochondrial DNA and chloroplastic DNA. So those are the types of DNA that we can cell. Because each and every cell in a mitochondria. And if it's a plant cell or other organisms, for example, like protists, or mitochondria, chloroplast. Chloroplast DNA yayona as a whole. So that's the location of DNA. Just to reiterate, it contains what? It contains nitrogenous bases that are base pairs. Base pairs meaning A, T. It's a magadi party. Amaba cross-nation. C o tlo phelana le motho wa hae G, T o tlo phelana le motho wa hae A o tlo phelana le motho wa hae T. Bona ke Ana le Tshepo, ho Molemo le Caroline e fella da so aba crossana. So those are the base pairs, and then they form this double helix structure. So you might wonder, man, so in this DNA structure it wrap around what we always say when it's about to get organized, it coils up. It's wrapped around the protein. It's in the histones. Okay, lo wrapper o matlapa so ne e bo wrapper the wool around you. Ne if let's say something that is organized. So re the histones, these proteins. Ne o ka re matlapa. Then purpose ya DNA ke ne. Purpose of DNA basically is to code for genes. Ne? It carries what we call the hereditary information. Hereditary information. Ne? Hereditary routing, we can pass genes from parent to offspring. This year we're going to use the word offspring a lot, not children. man are improving more life soft life re to offspring. So most of your DNA yona le yona are useless, are irresive. Next. Re bitsa non-coding DNA. I code is sip e teng shem. E teng e ntse a dilo fela so. Ne? And then ho na le e nngwe part of DNA ya hao. Ape DNA ke ba in a single nucleus. We re bitsa coding DNA. That's the useful DNA. Ke yona e codang nko e kima that you see ka mo generation from generation to generation. Papa o na le nko e kima, ntatemoholo wa hao, ntatemoholo wa hao, your forefathers and forefathers. Nko ela khalle teng. Hele le ba makhubela. Yeah, la ba itse ka nko e kima. So basically it comes from this coding DNA. So from this coding DNA, what is the main function of DNA? It controls functions of the cell. Ne? All functions of the cell, yeah, control mo hohle mo, ne? And then it regulates control the functioning of genes. Ne? It regulates the functioning of the genes. And then it passes on hereditary information. To pass on hereditary information, you pass on the big head, the ears, and the nose that your family has from one generation to the next. Ne? Good, and that is why in most families they look for those features. If you don't have them, so those are the main functions that DNA controller functions of the cell. It regulates functioning of genes and then passes on hereditary information. So what I want you to do for me as a form of exercise for ending this topic, go draw yourself a nice big structure of a nucleotide with the nitrogenous base adenine to it. And that will be the end of our lesson for now.